Hi everyone, welcome back. So today we're going to back up a little bit, and earlier in the semester we actually skipped a topic just so that we could keep our lecture topics and our lab topics in sync. So the topic that we skipped and said we would come back to it at the end of the semester is the cell cycle, mitosis, and meiosis. So that's what we're going to talk about today, and on the day of recording this lecture, it is currently Tuesday, April 7th, so today is Teddy's birthday. She is turning 4 years old, and she's doing what she's done every other day, and that is she's in the room that I'm recording in, and she's staring out the window. So she'll probably go back and forth between staring out the window and sleeping. She leads a very exciting life. So anyways, let's get started. So today we're going to talk all about when does a cell divide, why does a cell divide. So we're going to go through some definitions, and then we're going to talk about the cell cycle, and we're going to talk about mitosis, meiosis, and then how prokaryotes divide. And our overarching question that we're working on answering for this topic is this idea of how do we make more cells? So we're going to start with the cell theory. So at the beginning of the semester, we actually talked about what the cell theory is. So this will be a really quick review. And this question is asking us to think about which of the following is part of the cell theory and which one is not. So let's go ahead and walk through this together. So for A states that all living things are composed of one or more cells. That is true. B, all cells are about the same size and contain organelles. So that one is not true. So we know that cells are different shapes, different sizes. They all do have some sort of organelles. Not all of them have membrane-bound organelles, but that one is definitely false. And so we can kind of continue on and walk through the other ones. So for C, cells arise only by division of previously existing cells. That is true. And D, the cell is the basic unit of organization of organisms. Absolutely. So those are our three tenets of the cell theory. And today we're really going to be focusing on C. And so C talks about cells arising from previously existing cells. So this idea of omnivorous cellula e cellula. So all cells come from pre-existing cells. And we're going to talk about how that happens in today's lecture. So before we get into it, there's two questions that we're going to talk about. And that is, well, what do cells need before they divide? And why do cells divide? So go ahead and pause the video, brainstorm some ideas, and then when we come back, I'm going to go ahead and give you a couple of ideas to answer these questions. And then, as always, if there's something that I didn't mention, go ahead and leave it in the description or in the comments below. So let's start with the first question. What do cells need before they divide? Well, first and foremost, they need to make sure a new cell is needed. So although we are constantly making new cells, our body does a really good job of controlling and deciding if and when a new cell is needed. The cell itself has to grow larger, and so that's going to need a whole bunch of new biomolecules. So all of those macromolecules that we've been talking about, lipids, proteins, etc., we're going to need to make more of those so that we can have the cell grow larger. A big thing that we need to do is DNA replication. So we need to make sure that when those cells split, that each cell gets the right amount of DNA and the correct DNA as well. So we will actually have an entire topic that is devoted just to DNA replication. So when we finish up with the cell cycle, mitosis, and meiosis, we will actually move on to DNA replication. And this topic and the topics that we're going to talk about, so the entire unit that we're ending the semester with, are probably some of my most favorite topics to talk about. So I hope you all are really excited. And then, of course, when we make and when we copy DNA, we sometimes have mistakes. And so we have to check and fix any DNA mistakes. So we will actually end the semester with thinking about DNA mutations. We're going to do a case study at the end of the semester. And then for those of you that are going on to genetics, in genetics, you'll actually learn about some of the DNA repair mechanisms. So these are all things that cells are going to have to do before they divide. Now the next question is, okay, well, why do cells do this? Why do cells bother doing any of the division that we're going to talk about? First and foremost, growth. And that could be growth of the entire organism. That could be growth of the population. And so bacteria, of course, are single-celled organisms. Depending on the type of bacteria, anywhere from 1 to 10 bacterial cells is enough to make us really sick. So, for example, things like E. coli, any of those enteric pathogens, typically about 1 to 10 cells is enough for us to have violent throwing up and violent diarrhea, depending on the strain that you pick up. And so growth of the population as a whole, but of course growth of the organism if it's a multicellular organism as well. Repair. So if you get a paper cut, you fall, you get a scab, so on and so forth. We need to repair and replace the cells that are damaged. And then, of course, reproduction. So as we continue throughout this topic, although we're mostly going to focus on eukaryotes, we will come back and touch on prokaryotes at the end. The overall process is pretty much the same for all cells, and that's going to be copy the genetic info, separate the genetic info, and then separate the daughter cells. But we're mostly, like I said, going to focus on eukaryotes, but we will still touch on our prokaryotic friends. So let's start with a party fact, if you will. So this is something that we take for granted and we don't necessarily really think about. But what I want you all to do, and there is a right answer to this, but obviously you're not going to be penalized. I'm just curious to know, how many cells do you think that the typical human produces every minute? So go ahead in the comments below, tell me what you thought before I actually tell you what the answer is. So for those of you that picked C, 1 times 10 to the 8th cells per minute, you were correct. So every minute, on average, this is how many new cells our body produces, which is pretty amazing. So in part 1, most of this will be thinking about different types of definitions and getting ourselves prepped and ready for mitosis, meiosis, and for binary fission, which is how bacteria and archaea, how most of them will divide. So unfortunately for us, when it comes to this topic, there are a lot of words that start with C and they all sound the same. So we're going to talk about what chromosomes are, what chromatids are, and what chromatin is. So we're going to start with chromosomes. And we've actually been using this word and kind of throwing it around this semester without really fully defining it. I think we all have a definition of what it is, and I'm going to give you one, and then we're going to talk more about it and how it compares to our other C words. So I like to say that chromosomes are carriers of genetic information. And we'll talk about what chromosomes are made up of here in a second. But before we do that, let's do a compare and contrast of bacterial and eukaryotic chromosomes. And I had a spelling error. All right, so let's go ahead and let's do bacteria on the left, and we'll do eukaryotes on the right. So I encourage you to go ahead and to pause the video and think back to our first topic when we first began this idea of comparing and contrasting different types of cells and see what kind of information you can pull in. So for bacteria tend to have 1 chromosome, whereas eukaryotes have many chromosomes. The next major difference between bacteria and eukaryotes is that bacteria have a circular chromosome, whereas eukaryotes have linear chromosomes. Both bacteria and eukaryotes have double-stranded DNA as their genetic information, so anything that is considered alive all cells, regardless of what domain of life they are in, have double-stranded DNA. The only exception that we know of thus far to this double-stranded DNA being the genetic information is in viruses. So viruses break all sorts of rules, but other than that, any cell that you can think of always is going to have double-stranded DNA as its genetic information. And then last but not least, let's think about where the chromosomes are stored. So in the bacteria, they don't have any membrane-bound organelles, and so that chromosome kind of hangs out in the nucleoid, which is really just the area where the chromosome is found. It's not super organized. Whereas in eukaryotes, we have a double membrane that surrounds the nucleus, and that's where our chromosomes are found. So for the majority of this lecture, we're going to focus on eukaryotes, and we're going to ignore our prokaryotic friends until the very end of this topic. So we've defined chromosomes. We've said that they're carriers of genetic information. Now let's talk about another C word. Now let's talk about chromatin. So what is chromatin? So when we think about chromatin is really just a complex of DNA plus proteins. And chromosomes are made up of chromatin. So DNA does not exist by itself in the nucleus. DNA is actually wrapped around proteins, and you might be wondering, why do we have to have proteins? Why is DNA compacted? And that's a really great question, and it goes back to how much DNA we have. So if we were to take all of our chromosomes and we were to unravel the DNA and lay the chromosome from one end to another, we would have about 12 feet of DNA. And so it's kind of amazing that that much DNA fits into a nucleus. And so when we have DNA is wrapped around proteins, and these proteins are called histones, and these are further organized into a chromosome. So a chromosome is just made up of chromatin, and chromatin just refers to DNA plus proteins. So we're actually going to draw a model of what this looks like. So these are going to be our histones, and we actually get a bunch of histone proteins that come together. For this class, we're not going to worry about the numbers of them. If you take my upper level cell biology class with me, we actually get into a little bit more detail of what the histones do and why they're important. So we're going to have DNA that is wrapped around those histones, and I'm going to do my best here to try to illustrate that the DNA is double-stranded DNA. So we're drawing on the left from the most unwound, and then to the right, the DNA is going to get slightly more and more wrapped around the proteins, and they're going to get further organized. And so we start to get more and more condensed chromatin until eventually we kind of get a super condensed chromosome. So again, starting on the left here, here's our double-stranded DNA. All of the DNA is double-stranded. It starts to get wrapped around these histones, and then these histones start to further come together, and then we start to get this really condensed chromatin that is all just DNA and proteins, and then that further gets condensed into a chromosome. So our chromosomes can exist super condensed, like we will see during mitosis and meiosis, but they can also exist more unraveled and less condensed, typically in a nucleus. But our DNA is always going to be associated with proteins, so with those histones. And then when we have DNA that is wrapped around histones, we call this a nucleosome. And so oftentimes people refer to DNA wrapped around histone as beads on a string. And so chromosome is just made up of chromatin, and that is just DNA plus proteins. So now that we have covered chromosomes, chromatin, let's talk about chromatids. So I'm first going to give you the definition of what a chromatid is, and then we're going to practice using these 3 C words and how to apply them. So a chromatid is simply a double-stranded DNA molecule. So how the heck do we use chromosome, chromatid, and how do we know which one is correct? So let's go ahead and practice that. So let's draw one double-stranded DNA molecule, and you all will see me really struggle drawing double-stranded DNA molecules. And so we have one chromosome, and we can also say that we have one chromatid, because chromosomes are made up of chromatin, which is a double-stranded DNA molecule wrapped around protein. So we can say one chromosome, one chromatid. And when we copy the DNA, and we'll again have a whole lecture on how we do that, we then end up with one chromosome that is still connected, and they are identical. So this is still one chromosome because this is identical, and we have 2 chromatids. And those 2 chromatids are because we have one double-stranded DNA molecule on the right and one double-stranded DNA molecule on the left. The reason that we still call this one chromosome is because it is still connected and it has not yet been separated. And more specifically, when we have one chromosome that has been replicated, like we just drew, and we have those 2 chromatids, we more specifically call these sister chromatids. And sister chromatids indicates that they are identical. So after we have the replication of the DNA, we will have separation. And when we separate this chromosome and we separate those 2 chromatids, we then end up with 2 individual chromosomes. And at the very end of this, we now, because those sister chromatids are separated, we have 2 chromosomes, and we're back to 2 chromatids. So again, a chromatid is just a double-stranded DNA molecule. A sister chromatid refers to 2 chromatids that make up a chromosome and are identical. So let me go ahead and write identical underneath our sister chromatids. Okay, so we're almost done with introducing new vocabulary, and you'll actually see these chromosomes and these chromatids in the next example that we're going to talk about as well. So now we have to talk about homologous chromosomes and pairs. What the heck does that mean? So in this class, because we're talking about mitosis and meiosis, we're going to be talking about humans as well, and as well as other organisms that do sexual reproduction. We're going to be talking about the biological mother and the biological father. So I'm going to say biomom and biodad just to refer to the biological parents. And then when we talk about sex, we are referring to biological sex. So let's go ahead, and on the left, we're going to draw one chromosome pair. So for this chromosome pair, we are going to have one chromosome from the biological mother, and then we'll have one chromosome from the biological father. So let me try to pick 2 good colors here. There we go. I think those are a little easier to see. All right, so for a chromosome pair, what a pair means is that these chromosomes have the same type of gene encoded in them. So maybe this chromosome pair has the genes for eye color, for hair color, and for whether or not your ears are going to be attached. They might have different versions of those, but one comes from the biological mom and one comes from the biological father. So from bio mom and bio dad. So for these, we would say that these are 2 chromosomes. So a pair of chromosomes is 2 chromosomes. And right now in their unreplicated version, we have 2 chromatids, so 2 double-stranded DNA molecules. We can further call these 2 chromosomes that we have homologous. So homologous chromosomes are a chromosome pair. And again, chromosome pair just refers to the idea that they have the same type of genetic information on them. So same genes, but maybe different flavor of those genes. So this is going to, of course, be unreplicated. So what do these look like when replication occurs? So we're still going to have a chromosome pair. So they're still going to be homologous chromosomes. And let's draw. So we're going to have now from the biological mother, we have 1 chromosome, 2 chromatids. They are still connected. From the biological father, we have 1 chromosome that has been copied, and now we have 2 chromatids. So in total, after this replication, we still have 2 chromosomes. So we have 1 chromosome pair, 2 chromosomes, but now we have 4 chromatids because we have 4 double-stranded DNA molecules. And just like on the left, these 2 chromosomes were known as homologous chromosomes. That still holds true. So homologous chromosomes and chromosome pairs can be used interchangeably. We can actually add one more label to our image here. We now have, not only do we have chromatids, but we can be more specific, we have sister chromatids. So from the biological mother on the left here, that chromosome that was duplicated where we have 2 chromatids, those are identical, so those are sister chromatids. And on the right for the biological father, these are also sister chromatids. All right, so hopefully you all are still with me. I think this is one of the hardest parts of the online lecture stuff is that I can't see you all nodding along, or I can't see panicked looks, so I don't know if I've lost anyone or not, but just let me know. I know this stuff is confusing. There's a lot of terminology here to get through. Okay, we're almost done with terminology. The next set of terminology that we're going to talk about is this idea of haploid versus diploid. So the di from diploid means 2, and the hap from haploid means half. So when we have a cell or an organism that is diploid, diploid refers to a cell or an organism that has 2 copies of every chromosome, whereas haploid is going to refer to a cell or organism that has 1 copy of every chromosome. Okay, so if you can keep straight that di means 2, that means 2 copies of every chromosome. So every chromosome actually comes as a pair, and that will become really important here for our question that we have, and that is, how many chromosomes do humans have? So humans have 46 chromosomes, so 46 total. We can also say that we have 23 pairs of chromosomes. That 23 pairs goes back to the idea that 23 of our chromosomes come from the biological mother and 23 come from the biological father. So 23 pairs times 2 is 46 total chromosomes. So one of the things that we do in biology and in the field of genetics is when we have a diploid organism, we will say 2n. So another way to look at humans is to say 2n equals 46. So whenever you see 2n, that 2n tells you that that organism is diploid, and n tells you haploid. So again, not the most perfect definition for the N number, but for the sake of this class, to help us keep things straight, that's what we're going to go with. So when we say 2N, we know diploid, and for humans, we have 46 chromosomes. But we know that there's an exception to that, right? Not every single one of our cells has 46 chromosomes. Our gametes do not. So again, an exception to everything. So our gametes have 23 chromosomes. And so eggs and sperm, because they have half of the amount of DNA that is in our typical cells, half means haploid, they are considered haploid. So we would say N equals, and since our gametes have 23 chromosomes, we would say N equals 23. All right, so let's go ahead and let's look at something called a karyotype. So on this page, we have what we refer to as a karyotype, and a karyotype is a really fancy way of just saying a photograph of chromosomes. And typically, chromosomes are fairly uncondensed. Again, they're still associated with histones, but they're just not as highly condensed as they are for mitosis and meiosis. So when we take a photograph of chromosomes, when we do a karyotype, it's going to be after replication. And the reason for that is, as we will see when we talk about mitosis, the chromosomes will condense. So this is a karyotype of an organism, specifically a human, after replication. So you will notice in this image that we have 23 pairs total. So if they are all organized by numbers, they're falsely colored here, and you can see there's different sizes, there's different lengths of the chromosomes, there's different colors, which represents the different banding patterns. So each of our chromosomes have different genes, and it's been a really big interest to geneticists to map what genes are found on what chromosome. So that's why they're different shapes and sizes and different colors. So you'll also notice here that we have 1 through 22. And the reason for that is we have 22 autosomal pairs. And autosomal just means the not sex chromosomes. And so that's going to be 44 autosomal chromosomes. And then if we take a look here at this 23rd pair that we have an X and a Y chromosome. Thus, we would say that this individual would be biologically male. And so humans have 22 autosomal pairs and 1 sex pair, which is 2 sex chromosomes. So if we add those up together, we get 23 pairs or 46 chromosomes. All right, so now that we've had a couple of different definitions that have been thrown at us, let's go ahead and practice those. So here is an image of 2 cells, one on the left and one on the right. And what I want you all to do is to match the term to the appropriate arrow. So I suggest that you go ahead and pause the video and walk through this practice problem. All right, so if we start with A is pointing at a chromosome, and at this point we see one line. We can tell that this is an unreplicated chromosome. So 1 is A, whereas if we look at B, we see that X shape that many people think of when they think about a chromosome. So there we have 2 sister chromatids and 1 chromosome. So this would be B. And we can actually go ahead and add those labels. So on the left cell, we have 4 chromosomes and 4 chromatids. On the right, after replication, we have 4 chromosomes and 8 chromatids. All right, let's do another practice problem. So for this practice problem, this arrow is pointing to a circled part of a chromosome, and I want you all to tell me, based on what you know about DNA, which representation of DNA best fits the portion of the chromosome that is indicated by this circle. So hopefully you've taken some time to work through this one, and we talked about how chromosomes are double-stranded DNA, so we can cross off A because that is a representation of single-stranded DNA. And then on the left in that cell, we have unreplicated chromosomes. So one chromosome is equal to one chromatid, and we know that a chromatid is a double-stranded DNA molecule. So we have one double-stranded DNA molecule that is circled in red. Therefore, we pick the representation of DNA that most closely resembles one double-stranded DNA molecule. And you can probably guess what the next question is going to be. The next question is going to ask you about the cell on the right. So for the cell on the right, that arrow is pointing to one chromosome, but two sister chromatids, or two chromatids if you didn't want to be that specific. And we know that a chromatid is a double-stranded DNA molecule, and we have two of them there. Therefore, we have two double-stranded DNA molecules. So let me know if I lost you in the comments below. And what we're going to talk about next is, now that we have all of our definitions and we've practiced with them, we are now ready to talk about the cell cycle. So before we get into mitosis and meiosis, we first have to introduce the cell cycle. And the cell cycle has two main phases, and those main phases are interphase, and interphase is made up of three phases, G1, S, and G2. The second phase of the cell cycle is mitosis. So we're going to go ahead and actually draw the parts of the cell cycle, and then we'll label what falls under interphase, and we'll talk a little bit about what happens in each of the phases as well. And the G that we have, I'm going to write this up here, the G stands for gap. So we're going to start with G1, and G1 is a growth phase. So a gap phase, this gap phase is just a growth phase. The cell is growing, it's making more biomolecules, and within G1, we actually have a checkpoint. So this is our first checkpoint of the cell cycle, and in this checkpoint, the cell asks, do we need a new cell? And using signals from both the internal and the external environments, the cell will determine whether or not a new cell is needed. If the answer is yes, the cell will continue into the next phase. If it is no, it'll just kind of hang out and continue in that phase until it is either needed or until the cell undergoes apoptosis. So after our G1 phase, we have the S phase, which is the synthesis phase, and this is where we copy the genetic info. After the genetic information is copied, we have a second gap phase or a second growth phase. So this is our G2. And in G2, we actually near the end have another checkpoint. So this is checkpoint number 2. And in checkpoint 2, the cell wants to make sure that the DNA has been replicated. In addition, the cell wants to make sure, has there been any damage to the DNA? Did we fix the damage? Were there any mistakes? Have those been fixed? And everything that we have drawn so far, so let's go ahead and let's pick this yellow color. We're going to highlight everything that is interphase. So this part that is all yellow, so G1, S, and G2 is all interphase. after interphase is over, the very last step that we have is mitosis. So that's the M part, which stands for mitosis. And we will go into more details of what happens during mitosis. And we actually have a third checkpoint. So this is checkpoint number 3. And in this checkpoint, the cell is asking, are the sister chromatids ready to be separated? So this is what we refer to as the cell cycle. So cells kind of lead pretty boring lives. They go through this over and over again. And just to give you an idea of about how long this takes in a pretty rapidly growing cell, so this is going to be things like our skin cells, our intestinal cells, those tend to have pretty fast turnover. This entire cycle takes about 24 hours. So a cell will spend about 9 hours in the first growth phase, so in G1. It'll take about 10 hours to copy the genetic information, and then it'll take about 4 hours for that second gap phase and about an hour for mitosis. And again, that's going to be for a pretty rapidly growing cell. So now, based on what we have learned so far, let's go ahead and do a quick practice problem. So in this practice problem, we are going to figure out if a cell has 3 pairs of chromosomes, how many chromosomes will the cell have in G2. So again, I encourage you to go ahead and pause the video and try to work through this problem. So hopefully you've gone ahead and done that, and what we're going to do is I'm going to highlight the important part here. So the important part is that I am telling you that this cell has 3 pairs of chromosomes. So when you think of pairs, we should automatically be thinking diploid and thinking 2N. So now what we have to do is figure out how many chromosomes we have. And we're going to start with G1 and then we'll get to G2 in a second. So if we have 3 pairs, a pair is 2 chromosomes. And so we have 3 from the biological mother and 3 from the biological father, which means we have 6 chromosomes total. So let's go ahead and draw what G1 looks like and what G2 looks like. So we have 1, 2, 3 from biological mother and 1, 2, 3 from biological father. So in G1 we have 6 chromosomes and 6 chromatids. And to get from G1 to G2, we have to go through S, which is replication. And so we will end up with each of those chromosomes being replicated. And so we have 6 chromosomes, but each chromosome is replicated, so we have 12 chromatids. So again, just more practice in putting all these pieces together. So let me know if you got this correct in the comments below. So before we move on and talk about mitosis and meiosis, I first want to tell you the 2 types of cell division. And you all already know them. We've actually been talking about them without even fully defining them. And that is mitosis and meiosis. So for mitosis, this is going to happen in what we refer to as our somatic cells. So everything that is not a gamete. So our skin cells, neurons, eye cells, liver cells, so on and so forth. And mitosis is going to be cell division that results in identical copies. So we will have one cell that is diploid. So if it is diploid, I should say, and that one diploid cell will then copy and divide, and we will have 2 cells that are both 2N or diploid. In meiosis, this is going to happen when we make and produce gametes, and this is going to be a type of cell division that results in the reduction of chromosome number. So we go from one cell that is diploid to 4 cells that are each haploid. So what we're going to do next is we're now going to go through each of these types of cell divisions. We are going to start with mitosis, and day 1 of this topic will actually end at the end of mitosis. When we come back from day 2, we will talk about meiosis, and then we'll do some more practice problems. So before we get into mitosis and meiosis, using what you know thus far, I want you all to go ahead and circle the cell types that are likely to be created by mitosis. So recall that mitosis is somatic cells, and that's going to be cell division that results in identical copies. So skin cells are going to be produced through mitosis. Neurons will be produced through mitosis. Sperm cells are gametes, and these are going to be produced through meiosis. Blood cells can also be produced. And then for egg cells, these are also known as oocytes, and they're the biological female gametes, and they are produced through meiosis. So A, B, and D are human cells that are likely to be created by mitosis. All right, now we have all of the pieces and we are ready to talk about mitosis. Woo! All right, so we're going to start with a general overview, and I actually have 2 videos for you. So what I recommend you all do is actually pause this lecture and go watch the videos. So the top video goes through each of the steps, and so that will summarize a lot of the things that I will tell you about and the things that I will draw, but their pictures are much prettier than the ones that I'm going to draw are. And then the second link is a really cool one, and what scientists did was they fluorescently labeled the different parts of the cell, and so you can actually see the chromosomes compacting and you can see them moving, and it's a really amazing video. So I recommend that you all go ahead and watch those before you watch the rest of this lecture. So I'm going to assume that you have done that, and what we're going to talk about before we get into all of the details is just a general overview. So when people think about the stages of mitosis, you have probably heard or seen IPMAT. So sometimes people say IPMAT, IPMAT, IPMAT. There's different ways to remember it. So when I was in undergrad, I think this was an undergrad when I was taught this, we were taught IP mostly at 12. Again, lots of different ways that people have described how you can remember IPMAT. But it turns out that when we actually have mitosis, not all of these are part of mitosis. So let's talk about what IPMAT stands for. And what I'm going to do is anything that I write in blue will be a part of mitosis. Anything that is not in blue, so I'll continue using the black ink color here, that will be a stage that is considered to be outside of mitosis, but we're still going to talk about it for the sake of completion, because if you don't, mitosis doesn't make any sense. So the first phase that we have is interphase. Following interphase, we will then move on to the part that is considered part of mitosis. So we have prophase. M stands for metaphase. A stands for anaphase. And T stands for telophase or telophase. Tomato, tomato. And then after telophase, the last thing that we have is cytokinesis. And so what we will do here on the next page is we'll actually start with the blank page and we will draw the entire mitosis process together. But to give you a general overview of what happens, let's go ahead and start with a cell. And I'm going to make a pretty big nucleus. And we'll draw a problem with 2 pairs. So let's say that we have 2N, so we have 2 pairs. So if we have 2 pairs of chromosomes, that actually means that we have 4 total chromosomes. So we're going to have 2 from biological mother and 2 from biological father. And after we go through mitosis, so we go through interphase, mitosis, and cytokinesis, at the very end, we will have 2 daughter cells, and each of those 2 daughter cells will have 2 chromosomes that came from biological mom and 2 from biological dad. So we started with 2 pink, 2 orange, and each of the daughter cells will have 2 pink, 2 orange, and each of those daughter cells are 2n equals 4, and we say that these are 2 identical daughter cells. So now that we've got our general overview, we know what we started with, we know what we're ending with, we're going to go ahead and draw the whole process. All right, so we're starting with a blank page, and we're going to start on the left and kind of try to work in a circle here to represent the cycle. So we're going to start with interphase. You're going to see me drawing a lot of really bad, poorly drawn cells, but that's all right. I'm going to do our best here. And we're going to begin with interphase. And in interphase, we go through G1, S, and G2, and DNA is copied. And for this example, we're going to do an example of 2 pairs of chromosomes. So 2n equals 4. And we're going to keep our same colors from the previous page, so that pink and orange. And technically at this point, I'm going to draw squiggles. And it kind of reminds me of ramen in a bowl, essentially. And that is because our chromosomes are not yet condensed. They are copied, they are replicated, but they are not yet condensed enough to the point where we can see that they are replicated. So at the end of interphase, if we pass that checkpoint, our DNA has been checked, no mistakes, we are ready to go into mitosis. The first phase of mitosis is the P, and that is going to be prophase. So for prophase, we are going to see a couple of things. The chromosomes condense. So now at this point, if we were doing a karyotype, we would actually be able to not only do that, but underneath a microscope, we would be able to see those condensed chromosomes. And we start to have the breakdown of the nuclear envelope. And then we're also going to start to form the spindle fibers. So hopefully you recall, and when I'm editing this, I'll paste a picture in of our cytoskeleton lecture when we talked about this, but we talked about centrioles. So the centrosome is made up of a pair of centrioles and how those are the microtubule organizational center. And so in this stage, we are going to start to develop those spindle fibers from those centrioles. I'm going to draw our nucleus kind of with holes around it because that nucleus is starting to break down. Our chromosomes have condensed so we can see them. So we said that we have 2 pairs, or 2n equals 4. So we have 4 chromosomes, but 8 chromatids because they have been replicated. This cell is then going to continue into prometaphase. So prometaphase is not something that is typically part of the IPMAT, and that's one of the things when you have shorthand things to remember, you don't always, not every single letter actually makes it in there. So we could go from prophase straight into metaphase, but I like to include prometaphase because it's that phase between the two, and it kind of fills in the gaps of what happens. And during this phase, what we have is the spindle fibers will actually attach to the chromosomes. So at this point, our nuclear envelope is completely gone, and those chromosomes are going to, and the spindle fibers will attach to the chromosomes, and the chromosomes will start to move. And then the next part that we get to is metaphase. And the metaphase is the M part of that mat, and in metaphase, our chromosomes line up in the middle. So M, metaphase, M, middle. That's how I like to remember it. So we will get those chromosomes beautifully lined up in the midpoint of the cell. And of course, they're still connected to the spindle fibers. So the movement of those spindle fibers helps get those aligned. And this is to make sure that each daughter cell gets the correct number of chromosomes. The next phase that we have, and I'm going to draw this cell a little elongated here, is anaphase. And anaphase I think of as away, and in anaphase the sister chromatids separate. So those sister chromatids are separated, and there's a lot of experiments that have been done in terms of how the spindle fibers work, so how these microtubules are accomplishing that, and it's actually something that we talk about in my cell biology class. So if you're interested, we talk at pretty great depth about how that occurs in that class. So as those sister chromatids are separated, we start to begin to pinch off those cells, and we call this the cleavage furrow, and this is telophase. And in telophase, the chromosomes are at the polar ends and the nuclear envelope reforms. So let's go ahead and draw the nuclei reforming. And each of what will become the daughter cells should each have 2 orange and 2 pink chromosomes. And then last but not least, draw a small arrow here because I didn't leave myself enough room. We have cytokinesis. And in cytokinesis, we have the actin-myosin ring that occurs. And again, we have mentioned this before in class, so I'll go ahead and try to insert when I'm editing an image of that. And during this stage, our chromosomes begin to uncondense as well. So they're back to looking like ramen in the nucleus. And in cytokinesis, we have the division of the cytoplasm. And again, we here have drawn an animal cell. As a plant cell, we actually would have an extra consideration. So an animal cell has a plasma membrane that keeps everything together, but a plant cell, in addition to that plasma membrane, has a cell wall. And although we're not going to get into great depths about it, I did want to mention that the process for a plant cell is very similar. The difference is that during telophase and during cytokinesis, we actually have to make a cell wall there, or what we call a cell plate, because of course we need a cell wall when those 2 daughter cells are formed. And the way that we do that is actually using the Golgi. And so the Golgi will send vesicles, and those vesicles will have the pieces needed, that cellulose, to make the cell plate. So just something I wanted to kind of throw in there, that although we have drawn an animal cell, that for a plant cell, this is very similar. We just have an extra consideration. So now that we have gone through mitosis, we're going to do a couple of practice problems. So what I have in here is a summary of mitosis. So here is just a textbook image, and it has a cartoon image as well as some stained pictures taken by microscopes. And so this is in here for you all just as an extra resource. And what we're going to do, like I said, is some practice problems. So here on the right, we have a cell, and on the left, I'm asking you what part of the cell cycle you think this cell is likely in. So if we take a look at the cell, we have that X shape for our chromosomes, which means that they are replicated. So in this cell, we have 4 chromosomes and 8 chromatids. And so for these, because we have replication, we have chromosomes that are replicated, we can go ahead and cross off A, so the cell is definitely not in G1. We can cross off B because the chromosomes are not being replicated. They already are replicated. And that leaves us with the second gap phase, G2, or D, mitosis. This cell is likely going to be somewhere between C and D. So you could make an argument that because these chromosomes are already condensed, that we are at the beginning of mitosis or at the end of G2. So either C or D would be acceptable for this question. And let me know in the comments below if you have any questions on that. Okay, so the second practice problem that we are going to do is thinking about this cell that is in the upper left-hand corner. So this one right here, what would it look like if it was in metaphase? So this cell is, like we said, in G1. It has 4 chromosomes and 4 chromatids, at least in G1. And looks like it's got a green, a red, an orange, and a blue chromosome there. So when it goes through the S phase and through replication, what we'll end up with, let's see if I can match these colors up here, we'll have a blue chromosome that is replicated. The green is longer, so we'll have a green chromosome that is replicated, a long red chromosome that's replicated, and a short orange chromosome that is replicated. So at the end of G2 going into mitosis, we have 4 chromosomes or 8 chromatids. And so this question is now asking us about metaphase. So metaphase is where the chromosomes have lined up in the middle. If we take a look at our choices A, B, and C, all of those look like they have lined up in the middle. So let's go ahead and see if we can start crossing off some of these answers based on the number of chromosomes and number of chromatids that we need. So right away, if we take a look at B, those chromosomes are not replicated. So we can go ahead and right off the bat, we can cross this guy off. So that leaves us with A and C. If we take a look at C, all of those chromosomes are replicated and they're in the middle, but we actually have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes, and each of those are replicated, so that means we have 16 chromatids. And we just said that the cell would have 4 chromosomes and 8 chromatids, so we can cross off C, which leaves us with A. And in fact, A has 4 chromosomes, and it has 8 chromatids as well. So again, let me know if you have any questions on these practice problems. And this actually wraps up mitosis. And so when we come back in day 2, we are going to talk all about meiosis, and then we'll end this topic talking briefly about how do prokaryotes undergo cell division. So have a wonderful day, everyone, and I'll catch you in the next video. Bye, all.